I did my undergraduate degree at the University College London in medicinal chemistry, and during that period I did both chemistry, biochemistry, and pharmacology, and so it stoked my interest in the use of chemistry to develop drugs. I then proceeded to Oxford to do my PhD in organic chemistry. But the desire to use chemistry to develop drugs wasn't lost, and so I then proceeded to do a postdoc at Stanford University. But this time, I changed directions from organic chemistry, which is making molecules, to actually looking at DNA replication and how one can use small designer molecules to actually study how the body copies its genetic material. So after Stanford, I then moved to University of Maryland, College Park, where I set up my independent career in the general area of chemical biology. So for my independent career, I was interested in again using organic chemistry to look at biological problems, but this time I was interested in bacterial communication, how bacteria talk to each other to cause diseases, form biofilms. And so we set up a program where we discovered some really interesting molecules that can actually stop bacteria from forming biofilms. In the last few years at the University of Maryland, College Park, I started also looking at certain processes in cancer, specifically the role of G quadruplexes in the expression of some cancer genes. And so we started developing small molecules that could also inhibit cancer cell proliferation. And so at Purdue, my group is actually interested in two general areas. One is bacterial communication. We are still interested in not only developing small molecules to inhibit bacterial communication, but also to use the tools of organic chemistry to uncover fundamental pathways in bacteria and how bacteria responds to the environment. We also have a program in cancer, and so we are also developing new therapeutics that target kinases, G quadruplexes, and the ultimate goal is to have a small molecule that could be translated into an anti-cancer agent.